Welcome everyone, happy Friday. Thank you for joining us today for our Meet the DM Drug Developer series. Today, May 7th, we are talking with Biogen and Ionis Pharmaceuticals. As a reminder, the MDF mission, Care and a Cure, is to enhance the quality of life of people living with myotonic dystrophy and accelerate research focused on finding treatments and a cure. MDF's work focuses on support and education, research, and advocacy. A reminder about the resources and support opportunities available through our website, our toolkits and publications, including clinical care recommendations and guidelines, information about our virtual support groups and Facebook chats, our calendar of events and activities, as well as our Digital Academy. And the Digital Academy includes presentations and videos from past events or presentations, as well as it will include today's presentation. Okay, like today, the first Friday of each month up until September 10th, which is our annual conference, we will be meeting with different drug developers. We have one exception to that, and that is on Wednesday, June 9th. Because of the very high demand to talk with different drug developers, we added an extra session. It happens to be on a Wednesday, so mark your calendars. You can register for any of these sessions and submit questions for presenters in advance at myotonic.org-meet-dm-drug-developers. Similarly, on the third Friday of each month at 12 o'clock is our Ask the Expert series. So this month on May 21st, we are going to be talking about the heart and myotonic dystrophy. If you have questions about any relationship with the heart and myotonic dystrophy, please register for this event and submit your questions in advance so we can get them to our expert. They will attempt to integrate all of those questions into the presentation. So even if you are not able to make it live during their presentation, if you've submitted questions in advance, they will do their best to answer those during the presentation. You can sign up at myotonic.org/ask-expert-series. We have declared, a global alliance has declared International Myotonic Dystrophy Awareness Day on September 15th. Learn more about the Global Alliance and what you can do at myotonic.org/international-DM-day. As I mentioned, all the sessions are being recorded from the series. They will be posted to the Digital Academy. Give us a little bit of time to render the video and get it onto the site. But if you do have any questions for us about today's presentation or about any of the other recorded sessions we have posted, please don't hesitate to give us a call. The phone number for the office is here, as well as our email address, which is info@myotonic.org. Okay, you are here to meet today's presenter, Dr. Katherine Meilleur. She earned her bachelor's at the University of Maryland in biology and then pursued her master's program at the University of Pennsylvania as a second-degree nursing student. After completing her bachelor's in nursing, she worked as a neonatal intensive care nurse at the Hospital of the University of Pennsylvania while finishing the Master's of Science nursing requirement. Dr. Meilleur completed the Master's in Science program as an acute chronic pediatric nurse practitioner and worked in the newborn nursery of HUP while attending Johns Hopkins University to complete a PhD in nursing. Dr. Meilleur's interest in international health and genetics led her to do her dissertation with a neurologist at the National Institutes of Health who had a partnership in Mali, West Africa. There, she identified a novel genetic locus for hereditary spastic paraplegias. She also developed a scale to test knowledge and attitudes of Malians before and after receiving genetic testing for the first time. After a postdoc at the National Human Genome Research Institute, she became a staff scientist in the National Institute of Neurological Disorders and Stroke, and then a principal investigator in the intramural program of the National Institute of Nursing Research. She studied clinical outcomes in pediatric neuromuscular diseases and performed trials to develop treatments for these conditions. Dr. Meilleur and her family moved to Boston in 2019, where she now works for Biogen in the clinical development unit as a principal clinical scientist. There, she continues to design clinical trials for neuromuscular diseases with a significant focus on myotonic dystrophy type 1. Okay, it is time to meet Biogen and Ionis. In order for you to send questions to Dr. Meilleur, please use the chat box in the drop-down menu. Select Send Question to Staff. That's the only way that she will receive your questions. So chat box, drop-down menu, select Send Question to Staff, and then she will receive the question. Okay, thank you all for joining, and welcome, Dr. Meilleur.
Thank you. It's my pleasure to join you today, and I appreciate this opportunity to share an update from Biogen and our partner Ionis on our drug development in DM1. Today we'll be talking about antisense oligonucleotides, or ASOs, as a potential treatment for DM1. First, I'll talk about who we are, Biogen Ionis, and why an ASO may help in DM1. I'm also going to address the patient manifestations and main symptoms, why we think an ASO may be effective for muscle symptoms, but also for the central nervous system. And then I'll also be talking about how we would measure if a muscle or CNS ASO is working, and provide a summary of where we are. I am an employee, by way of disclosure, I am an employee of Biogen and do hold stock in Biogen. Biogen is a company that's been around since 1978 and has the goal of defeating devastating neurological diseases. It's headquartered in Cambridge, but it has 39 office locations across 34 countries and approximately 9,000 employees in research and development, manufacturing, and general and administrative services. Ionis was organized or founded in 1989, about 11 years later, and pioneered a new platform for drug discovery and development based on RNA-targeted therapeutics. And it's located in Carlsbad, California, and is comprised of 800 employees spanning the same units. So what is an antisense oligonucleotide? Basically, an antisense oligonucleotide is a small piece of DNA or RNA that can bind to a specific molecule of RNA, and this blocks the ability of the RNA to make protein. So we know that the central dogma of molecular biology is DNA makes RNA makes protein. If we can block the RNA from functioning, if it's mutated, as in the example of DM1, the case of DM1, then we can block the production of the protein that's malfunctioning. And this is a better picture of that with more details. So here's your DNA in the nucleus, that the gene or the genes in the nucleus that are comprised of DNA. And these are transcribed into mRNA. And the antisense drug here in purple binds to this single-stranded mRNA. And this involves several steps where the antisense oligonucleotide binds to this target and then degrades this toxic mRNA. And so you get the destruction of the toxic RNA that has the mutation in it, and you get less RNA, and therefore the protein is not made, so you get less protein. And this applies to DM1 because of the toxic mRNA in DM1. As you know, there are the CUG repeats in the mRNA. And you can see in white here the cartoon of the mRNA, and this is misfolded in the case of myotonic dystrophy. And if you treat this with an ASO, it will degrade this RNA. And then the MBNL protein, which binds to this misfolded mRNA, is released and can function normally and cause correct splicing of downstream gene transcripts or RNA. And we can use this technology to address muscle symptoms. So previously, Ionis and Biogen have already partnered to do a phase 1/2 clinical trial. And this included 48 DM1 patients in a multiple ascending dose study. And you can see here on the right the design of the trial. So patients were screened 4 weeks in advance. They came in for their first visit and had a muscle biopsy, which is the red star. They then got several loading doses of the drug in week 1. And then we had them have 6 additional doses weekly over 6 weeks. We then performed a muscle biopsy after all doses were administered and had a follow-up evaluation period. And we also did various functional assessments like timed function tests. And this was performed across multiple centers across the US. And we tested 5 different doses from 100 up through 600 milligrams. And the objectives of this trial were really to look at safety and tolerability of this ASO. We also had secondary objectives of evaluating pharmacokinetics of the drug, how the body absorbs the drug by looking at blood and urine, and also muscle tissue effects. And we looked at biomarkers and clinical outcomes as well. So for the primary objective, we looked at lab values, echocardiograms, how the injections were tolerated. For the secondary objectives, we assessed how fast the body breaks down the drug and how fast the drug distributes throughout the body. We also looked at splicing changes in muscle. We know that splicing of certain gene transcripts is abnormal in muscle, so we looked at that. And then we looked at clinical outcomes like myotonia, strength, motor function, and patient outcomes, patient-reported outcomes. Unfortunately, this drug did not reach the target concentration of 10 micrograms per gram in the muscle. So you can see here that as we increased doses, we got closer to the target of 10, but we really did not reach the target. Here, the upper limit is 8, and over the average dose for this group at 600 milligrams was 3.1 micrograms per gram of muscle. So we were well under our target of 10 micrograms per gram. And based on this clinical data, we know that we need a 5 to 10-fold increase in either the concentration of the drug or the potency of the drug. And so the question is, how can we achieve that? And there are some newer methods for drug delivery to target organs, such as liver here. You can see that there's a receptor in liver hepatocytes, and using GalNAc conjugation, this enhances drug delivery to liver by 30-fold. So if you look at this ASO that Ionis developed for another indication, when they conjugated this GalNAc to the ASO, they found that the effective dose, or ED, went down to 4 milligrams per week from 122 milligrams per week. So this conjugation method works in liver, we know that. And the company Takeda also identified that we can do a similar conjugation using transferrin receptor to enhance delivery to muscle. So transferrin receptor, also known as CD71, is expressed throughout the body, including in muscle, and it's recycled. It's used over and over. And if we can conjugate our oligonucleotide using an antibody to this transferrin receptor, the transferrin receptor can deliver that to the muscle. And we know that other companies are working on this. So we're considering this as well. You can see here, for example, that knockdown of the target gene here, beta-actin, in muscle in the Takeda paper was improved with this conjugation to the CD71 or TFR. We're also interested in addressing CNS symptoms. So we're working on the CNS aspect of the disease. We have experience with other CNS diseases and therapies that work for them, such as Spinraza for spinal muscular atrophy and tofersen in phase 3 clinical trials now for ALS. As you may know, ASOs for CNS are delivered via lumbar puncture. And this, we identified the need to address CNS symptoms through two really important pieces of work that were done by the Myotonic Dystrophy Foundation and also the Marigold Foundation, which showed that when these foundations asked the patient community what are the most important symptoms, in addition to muscle weakness and myotonia, patients' lives are really affected and sometimes more affected by symptoms of excessive daytime sleepiness, fatigue, and cognitive dysfunction. So we take that very seriously at Biogen and Ionis, and we want to see if we can address these patient concerns. So how would we measure if an ASO that we develop for either muscle or CNS works? And this is why we need biomarkers and clinical outcome measures, because when we can measure that a biomarker works, such as a splicing biomarker, or when we can measure that a motor function improves or splicing becomes more normal, this lends support to the fact that we're really improving symptoms and affecting the activities of daily living of patients. And so we can then document this with these improvements in the biomarkers and clinical outcomes. And this is what gets us to a disease-modifying therapy. And there's been a lot of work showing that biomarkers improve the probability of success in clinical trials. So what this graph is showing in yellow is the percent of studies that have had, the percent of studies that have succeeded or are likely to succeed. So up higher is good on the y-axis, with and yet without biomarkers in yellow and with biomarkers in dark gray here. And you can see that in each phase of drug development, 1 to 2, 2 to 3, 3 to new drug application, and all the way through to approval, having biomarkers increases your probability of success. So that's really why we need to collect muscle, blood, urine, CSF. And we did this in our first study. We collected muscle biopsy samples from the 48 patients, and we looked at the splicing. We know that that muscle-blind protein causes abnormal splicing due to the CUG repeats in the RNA. And so what we found was that as we went and increased the dose, there was a hint that the higher doses improved splicing and restored splicing to normal, but we only were able to get a sense of this at the highest dose in a few patients, so it was not convincing, and a lot of that was due to the fact that we did not get that 10 micrograms per gram drug exposure in muscle that we wanted. But we have experience from that trial for the future. We also looked at symptoms of myotonia and muscle weakness. We can use those again in our future study to see if myotonia improves, to see if measures like the 6-minute walk test improve, or other measures of motor function. Now, it's a little more challenging to measure biomarkers and outcomes in CNS. And this is because there are a lot of obstacles to getting biomarkers from the CNS. So it's difficult with brain or with cerebrospinal fluid, CSF, to be non-invasive, to have it actually work or be operationally feasible, to repeat these measures so we can check if it's stable and reliable, and to see if these biomarkers respond to treatment. This is really challenging to do. That's because there's limited availability of the tissue. We can't really look at CNS effects from blood or saliva or urine easily. And so it becomes invasive. We have to get CSF from a lumbar puncture, or we have to do imaging, which is expensive, and we don't always have the appropriate imaging for all disorders. And of course, the brain is a very complex and heterogeneous organ. There are also few preclinical models that have a good CNS involvement that would teach us a lot about whether or not we're making a difference with the drug in the models. And so despite these challenges, we are working to identify either protein or splicing biomarkers that we can use in CSF. So we need to find robust and reliable biomarkers in the CSF and that respond to treatment. And we do have data from muscle to hint that this should be feasible, as I showed you. Now, with clinical endpoints, Dr. Janet Woodcock from the FDA has pointed out, and I quote, "It turns out that what is really bothering the patient and what is really bothering the doctor can be radically different things. Patients are true experts in their disease." So we really want to incorporate the patient perspective and also look at multiple aspects. As you know, there are multiple CNS aspects in this disease. Cognition, such as executive function, can be altered, but patients also complain of sleepiness and fatigue. So we need to cast a wide net and see what we can change, and that's challenging. We have to kind of address these obstacles and these challenges. We've established biomarker partnerships with several researchers, academic researchers in the US and Canada and Italy, and we're really looking forward to working with them and finding a way forward. So potential biomarkers could be RNA, DMPK. RNA DMPK is the gene that makes the protein, and we're also looking at genes that are alternatively spliced. So in summary, the drug development process starts with research and discovery and goes through preclinical testing and trials to regulatory review and approval. And then once a drug is marketed, there's continued surveillance of that drug. And so we are here, we've started clinical trials. Several different companies are in this stage. So we've kind of gotten to determine the therapeutic strategy and design ASOs, and we're working on showing they work in animals. So we need to identify, since our last drug did not have sufficient exposure in muscle, ASOs that do have that. and also other ASOs if we want to address CNS symptoms. And so as we test these for safety, we will then be able to move into human trials again, do phase 1, 2, and 3 trials. Hopefully these will have better efficacy, potency, and we will gain agreement from regulators. And then we will continue to monitor as a community with the regulators for safety of these in the future. So drug development is a cyclical process. We've talked about how ASOs could work in DM1 by addressing the exact problem in DM1, which is the repeats in the RNA that result in toxic nuclear foci and binding of MBNL to the toxic nuclear foci and resulting in altered splicing. And we're seeking to address both muscle and CNS symptoms by restoring normal DMPK RNA and downstream gene function. To do this in the clinic, we're developing biomarkers and selecting clinical endpoints and getting ready for a potential therapy for patients. And whatever, even if the trials do not work as we've seen in the past, we know this is a cyclical process where we learn more each time, and we will eventually succeed. I just want to acknowledge all the patients and their families, for those, especially those who participated in the last trial. Thank you for continuing to be interested and involved and supportive. We want to also acknowledge all of the sites from the last trial. In particular, from Ionis, I want to thank the team and especially Lori Mignon for her help with this talk, and especially the team at Biogen as well, and Gershom Dent as well for her assistance with this talk. And with that, I will take questions. Thanks. So the first question I'm seeing is, when do we expect the next clinical trial? So this is a really difficult question to answer, and I can't give specific timelines, unfortunately. It depends on a lot of factors, but we are working really diligently to do this as soon as possible. Let's see what some of these. We are another question I'm getting is, how close are you to collaborating on another trial? And it's been a couple of years, and why did it take so long? So it's actually very challenging to develop drugs. It takes a lot of work, both in vivo and in vitro cell culture, as well as in vivo animal models. So we are working as fast as we can, but I know that that seems to be taking a long time, and we already are collaborating together again. Are we aiming for a single drug, CNS and muscle, or 2 different drugs? So ideally it would be 1 drug. If that's not possible, it will have to be 2 different drugs. Are new conjugated ASOs being developed for muscle targeting to improve the target knockdown? Yes, we are considering this approach. Are you aiming for a single drug? I answered that. Is there a reason 600 milligrams is the high end for testing? So this is related to the previous study. Yes, we did see some side effects at that dose range. So actually 600 milligrams may be even too high. Some patients had a low platelet count at that dose range. Seems to be the RNA is a hot new method, just like COVID vaccine. Yes, RNA is definitely a newer approach for drug therapies. Ionis has been working on for a long time. How long until you commence trial in humans? That I am unfortunately unable to answer. Since the Pfizer COVID vaccine was developed using mRNA, has this helped your mRNA studies? No, my understanding is they're using a slightly different approach. Was the DMPK-RX drug in the last clinical trial have similar results in patient and animals? In animals, it showed more results than in patients. In patients, we did not have enough drug get to the muscle tissue, unfortunately. When do you expect the next clinical trial? Can't answer that one, unfortunately. Will the drug reverse DM1 or slow the progression? That's a really good question. So because DM1 is slowly progressing, we would like to possibly see stabilization, possibly improvement, but we won't really be able to answer that until the trial.
Thank you so much, Dr. Meilleur. I had a couple of questions that came to me, if you don't mind, that I could ask you, just a couple of them, and it looks like maybe one other one came in just a second ago. Some folks have participated in the last trial, and they're wondering if they'll be eligible to participate in your upcoming trial, and if not, or will they also be able to participate in another company's trial?
Yeah, that's a great question. So each trial always has its own set of inclusion and exclusion criteria. So in all likelihood, you would be able to, certainly you would be able to be screened, and we would evaluate whether you fit the inclusion criteria for our trial. So you should certainly, you know, once we start recruiting patients, certainly respond, and we can evaluate that. For other trials that other companies run, it's the same case. So they will have their own inclusion and exclusion criteria, and you should certainly respond to their recruitment as well. If they're running at the same time, we cannot have a participant in both trials, because then we won't know which, if one of the drugs worked, we won't know which one it is, right? So they will confuse, the results will be too confusing to filter apart, tease apart. So you would just need to be in that one trial that you decide to be in.
Okay, thank you. Another question that came in was, the drugs that you're developing now, particularly for people who were not familiar with your clinical trial, is it a drug that people would take orally or through an IV? Is it something they would need to take every day, every week, eventually? Sort of what will that look like for people?
Yeah, so that's a great question. Right now, we don't have any drugs available orally with this technology that we're using. So the ones we're developing would either be IV or possibly by lumbar puncture intrathecally, or they could be subcutaneous. And so right now it's probable that you would not be able to take them at home orally. That's something in the future that may develop, though.
Thank you. A question that comes sort of based on what we've been seeing from the COVID-19 vaccines, which seem to get approved for emergency use very quickly. Do you think that that may have an impact on clinical trials for other types of drugs moving forward? Do you think it will take fewer years to get approval?
Yeah, that's a great question. So I think that the experience with COVID has definitely pushed the clinical trial development pathway into this fast forward motion. The difference though with a disease like COVID is that there are thousands of people who have it and who could be in a trial. It's more difficult in rare disease. So yes, potentially the drug development process could start moving faster, but with rare disease, it may be harder to move that fast because we have to find all of the patients. So it's slightly different from COVID in that way.
Okay, thank you. Your question came in, do you still go to phase 1 safety trial with a new ASO, or will you be able to go directly to patients?
Yeah, so with ASOs, we are able to go directly to patients in like a phase 1B.
Okay.
So skipping the healthy volunteers.
Okay, thank you.
In most cases, again, it may depend on the specific drug, but in some cases that is feasible.
Okay. It's really clear it's difficult to determine when you might be in a human trial. But the question is sort of, can you try to gauge, is it, are we talking like a year, 3 years, 5 years? Just to try to determine if, it's around the corner, but we know it's in the future, or is it really far ahead?
Yeah, unfortunately, I'm not at liberty to disclose that. So although we have some internal timelines, we're not allowed to disclose that yet.
Okay, fair enough. Question that came in, are ASOs and DMPK-RX the same therapy?
So DMPK-RX is an ASO. It's one ASO that we used in the previous trial. And then ASOs in general are, the whole category of drugs. So that was one ASO that we used in the past.
Great. I think that was the last question that came in. I'll do one last shout out for questions. If anyone has an additional question, you would type it into the chat box, and in the drop-down menu, select send question to staff, and it'll pop up for us so that we can take a look. Okay, one question. So will the same ASO be used for the next trial, just a different delivery method?
Yeah, that's a great question. I don't think I'm at liberty to answer that one either.
Okay, no problem. Okay, I think that might be it. So this is actually really exciting. We're grateful that you could be with us today and share the progress, remind us of, what has happened with, one of the first and most exciting clinical trials that has come to DM. And we're hopeful that, lots of new things are happening and there's something in progress, even if, we're not at liberty to share, you're not at liberty to share with folks at this point. I do think that that's very hopeful. We have over 40 companies at this point investigating drugs and treatments for DM, and to know that you have continued your work and, you're trying potentially a new avenue is just, it's reassuring, and it's very, very exciting for the community. So thank you for sharing with us today about the progress that you've made and what some of the plans might be for the future. And we hope that you will join us at the conference in September and maybe give us another update about where things might be.
Yeah, and thank you for all you do for the patient community, Tanya, and to all those who are listening. Thank you for your participation today.
Fantastic. All right. Thank you. Have a wonderful weekend, everyone. Take care.